Hello, I'm Seulgi Kang in Sungkyunkwan University, and advisor professor is Seulgi. First of all, thank you for the KSBNS and GISTAR for inviting me. I'm going to talk about metabolomics investigation in depression model. Depression is a huge health concern because of heterogeneous pathological mechanisms and ineffective treatment and diagnosis. Notably, women are clinically twice as susceptible to depression compared to men. So far, many studies have been conducted in males. Studies on sex differences-related depression is insufficient, so in this study we aim to the possible metabolic pathways and biomarkers associated with sex differences in depression through metabolomics investigation. Maybe the audience who attended this workshop is now familiar with metabolomics research, so I'll give a brief explanation. Omics analytical techniques, including genomics, transcriptomics, proteomics, and metabolomics, have been introduced the detection for gene, RNA, and proteins, and metabolome, respectively. Among them, metabolomics is an advanced technique provides metabolite concentration in biological samples. Metabolites are the end products of complex cellular networks, and they can influence via feedback loops. There are two main approaches, namely non-targeted and targeted metabolomics. Targeted metabolomics is a hypothesis-driven, and it focused on quantitative measurement of metabolites and their flux. In contrast, non-targeted metabolomics is a hypothesis-generating, and therefore is the most common choice as a first step to concept and monitor the broadest range of metabolites. Professor Jie Zhang's lab established depression models and provides mouse samples. We employed a subchronic variable stress model, which is an abbreviated version of chronic mild stress, known to be more vulnerable to depressive-like behavior in female. Another stress model is an acute unhappiness model, which depression was induced regardless of sex. The flows of targeted metabolomics is as follows. First, experimental sets is 4 independent sets, and non-targeted and targeted metabolomics is in blood, whole brain regions, and liver were analyzed by LC-MS and GC-MS. In the analysis step, data pre-processing were conducted to obtain each data set. In the post-analysis, statistical analysis, identification of metabolites, and interpretation of metabolic pathways were processed. Let's take our results. PCA score plots as unsupervised analysis and OPSDA as supervised analysis provided the metabolic profiles of the plasma and whole brain were different depending sex and stress. The features with the variable importance in the projection value over 1.0 and p-value and q-value lower than 0.05 in statistical analysis were considered as differentially expressed metabolites. Altered metabolites were identified in female only. This table showed the differential metabolite number obtained from the discovery set. As interpretation of altered metabolites, glycolysis path pathway, ketogenesis, and lipid metabolism were disturbed in female after stress. These mechanisms suggested that there is a problem with energy metabolism. Furthermore, the level of corticosterone was elevated in female specific. So, only females displayed disturbance in glucose metabolism and HPA axis regulation. Diagnostic biomarker panels consist of 3-hydroxybutyrate, myoinositol, serine, and glycerol. 3 prediction models were constructed using support vector machine, random forest, and logistic regression. This model showed a poor discriminant power for male mice, but good prediction for female mice. Our results suggest that female-specific biomarkers were developed. Until now, it has been a result of SCVS. I will show about acute stress model. In this model, the corticosterone level was elevated in both sexes, but differential metabolites associate energy metabolism, long-chain fatty acid, neurotransmitter, and hormone precursor amino acid were different by sex after acute stress. From the result of research conducted, the SCVS model reveals the sex difference in the metabolic changes upon stress. Our results finding provide a novel insight into vulnerability to acute to subchronic and guide the development of personalized pharmacotherapies. I would like to thank to Professor Jong-mi and our lab members, especially thanks to Uni Kim, Ji Min-nam, and Professor Ji Hae-jung for collaborating with us. Thanks for listening.